Welcome back to the FARA interview series where we are taking you behind the scenes at the 2019 International Ataxia Research Conference. We are here with a couple of the founders of the field. We're so honored to be here with you guys.
Yeah, thank you very much.
And why don't you two introduce yourselves first of all?
Okay, so I'm Helene Puccio. I'm a research director in INSERM in the French National Research, part, and I work on FA from mouse models, cell models, all the way to getting treatment.
And so, Ellen, your lab is solely devoted to FA, right?
My lab is 3/4 solely devoted to FA. Yes.
Okay, sounds good. Thank you.
So I'm Massimo Pandolfo. I've been involved with FA since the 1980s. So I'm just one of the seniors here in this group in different parts of the world. The main accomplishment was back in 1995 when I was in Houston and we identified the FA gene and mutation. It was a large collaboration. I cannot take all credit for that because there was important contributions from actually Michel Koenig from the lab where then Helenes went and from other groups in other parts of the world.
But that paper came out of your lab.
That paper was had authors from 4 different labs.
Okay.
So it was really a collaboration.
Okay.
This word that is kind of the magic word.
Yeah.
Collaboration.
Right, exactly.
Like in all things.
Right.
We wanted to talk with the FA community about gene therapy. And maybe to start, can we define gene therapy as a big umbrella term that talks about a lot of different pieces. Can you give our audience an idea of, in a general sense, what gene therapy is, what that big umbrella means?
So gene therapy is modulating genetic material. So it's bringing in a medication that is a genetic type of material. And there's different ways of doing gene therapy, as we talked about. And you said you guys have already talked about some of the other type of gene therapy being in the FA field.
So for example, ASOs.
ASOs,
we talked about that
yes.
with Dr. Corey and
Yes.
Dr. Naparella. That's a form of gene therapy.
So that's a form of gene therapy. So it's bringing in genetic material as a medication. So the traditional, the more traditional gene therapy is gene replacement.
Mm-hmm.
In a genetic disease, when you have a gene that is missing or not expressed enough, as in FA, one of the easy thing on paper is to just say, well, why don't we just bring the gene back? And that's gene replacement therapy.
And when we talk about gene replacement, are we talking about adding a functioning fragment of a gene to a cell to see if that gene transcribes whatever protein we're looking for? Are we actually talking about, I think everyone's heard of CRISPR-Cas9, snipping pieces out and actually plugging a new piece in?
So you can use CRISPR-Cas to snip out and put in a new gene, or you can use CRISPR-Cas to actually snip out the expansion. You can do that in models right now. You can't do that in humans yet. Here we're talking about just bringing in a piece of the gene, a piece of the frataxin gene, and then hoping that it's going to be expressed in the cells. So we're not actually modifying directly the genome of the person.
Okay, let me see if I can get this, and tell me when I'm wrong. Inside of every cell, there's a nucleus, there's mitochondria all over the place, and what you're talking about, and inside the nucleus exists the entire strand of our DNA. Little parts of that DNA break off, and they leave the nucleus into the cytoplasm, and they're read by special devices called a ribosome, and theoretically, if everything works right, that ribosome produces a protein or whatever else we're looking for. What you're talking about is taking a little snippet of DNA that's the correct snippet, that doesn't have a GAA repeat, and sticking that into the cell in hopes that the ribosome reads that instead of the triplet expansion version.
Yeah, so you're just missing one step.
Okay, just one.
One simplified step. So it's not a snip of the DNA that comes out. It's the DNA gets copied into what's called mRNA and transcribed, and actually that's the copy part is what's defective in FA because of the expansion it cannot be copied correctly.
Okay.
And so then you don't have enough of that copy that goes out to be translated into a protein. You agree? to
I do.
explain that this way. So what we're doing is taking indeed a little piece of DNA, of the frataxin DNA, that does not have expansion, that we bring into the cells using viruses. That little copy goes into the nucleus, does not get into the genome, but goes into the nucleus, and then uses the machinery of our body to make a copy in the RNA, and then to be able to go and be translated by the ribosome to be making protein in a cytosol, and then to be important to the mitochondria, because that's where it works.
Okay. And that's what we're looking for, is we're looking for something to tell the ribosomes, make frataxin and get that frataxin into the mitochondria so the cells can work.
Yes.
All right.
Beautiful.
That's why I understand that you work with gene therapies as well. Can you describe your work?
So, yes. First of all, since my animal model is an advanced primate, Homo sapiens, I haven't done yet.
That's us.
I haven't done yet any experiments, but of course we are very much building on the work that has been done in Helene's lab, that has clearly given the proof of principle that this may work. And of course, this is not the only option. And here I come to the other magic word we already said, collaboration. And the other one should be option. When we will have a treatment, there will may be other ways of treating this disease and of getting the frataxin that is not there. For instance, by activating the frataxin gene, which is repressed by the GA repeat. And it would be really nice to have these different types of options, like giving the gene via gene therapy, like activating the endogenous gene, also via gene therapy, or with other ways, more kind of traditional pharmacological ways. I think that's very important. I think gene therapy offers a very interesting perspective for a number of reasons. I mean, this is really a disease in which the cause is not making enough of something, and you can introduce a piece of genetic information from outside that codes for that, and you can kind of get to normal levels. Of course, you have to be careful. We haven't talked about yet the risk of making too much of a good thing, for instance, too much of frataxin. That is part of all the technical hurdles that need to be overcome before we have a treatment to offer to our patient.
So we've come so far in gene therapy, and we truly are getting there. What are some of the hurdles that you could talk about? A couple more hurdles maybe that you might be able to identify that before we get there.
Well, I mean, the main hurdle is to have a vector where we call the gene therapy drug is called a vector that contains the piece of genetic information that we want.
The delivery mechanism.
The delivery mechanism, exactly, because we know what we have to give. We have to give a kind of a short copy of the frataxin gene that has all the information to make the protein. Now, how do we do that? And to do that, we usually put this piece of DNA inside a shell, and this shell is a macroscopic thing that has to be able to go where we want it to go. And where do we want it to go? In FA, we want it to go in a number of places. And FA is really a systemic disease. I mean, it touches the nervous system, but both what we call the central nervous system and the peripheral nervous system. It touches the heart, as many know. It may also have some effect on muscle, which is not really a myopathy, but may have an effect in the capacity of making energy, may have an effect on the cells that make insulin. So you really would like to have it as diffusely as possible.
Everywhere.
Spread. But this is problematic because, of course, how do we give this vector? We give it, for instance, intravenously. If we inject it locally, it will go locally. If you make a systemic treatment, we have to make sure that from the blood it can go to the target organs. And this is particularly for the central nervous system. This is complicated because the nervous system is protected by what we call the blood-brain barrier. Blood-brain barrier is the fact that it's particularly difficult for whatever it's in the blood to diffuse into the brain. It's more difficult to diffuse into the brain than to diffuse into, let's say, the liver. And of course, this is also to protect the brain from a number of offenses, including viruses. And that's a virus. What we're going to do? I mean, modified virus is probably it's not the only option, but it's the option that's being explored currently.
Most people,
Yeah, so we needed to have a virus that can pass the blood-brain barrier, and there are some viruses that can do that.
I understand manufacturing is a hurdle as well. Can you guys talk a little bit about that and what that means?
So manufacturing is making the medication, and this is a biological medication, so it's not just making chemistry.
Mm-hmm.
You know, you actually have to grow these. You need cells to make these grow. And you need to go from what we're saying, you know, whether it's from a mouse to a person, where, you know, or even from what Massimo was saying, from an SMA baby to an
Sure.
adult. I mean, so you need to make enough so that you have enough material to be able to target all the tissues that you want to target. So it's the amount you have to make.
When we're talking about scale, how much does a mouse weigh?
20 grams.
And how much does a human weigh?
A small one,
depends.
50. Depends. Between 50.
Hey, slow down there,
50,
Kyle.
Between 50 and 100 kilos. Depends who you are.
So that's quite a big difference. And so we're talking about a large amount of drug, of these viruses that you're talking about that we can administer in different ways.
Yes.
Well, I mean, 20 grams to 20 kilos is 1,000-fold.
There we
If
go.
you think about it, and the 20 kilos is just a fraction of my weight.
You have both been, and we're incredibly lucky as a community for this, you've both been in the FA space for a very, very, very long time. Massimo, you were on the paper that described the gene. Helene, you've been working in this field, doing amazing work and publishing your own papers. Why did you choose FA? And maybe as a corollary to that, we're at the International Ataxia Research Conference. Can you talk a little bit about the collaboration that you have with the European organizations and FARA and how this all works together?
So why am I working on FA? So
Yeah.
I started working on FA just as all of us, I think, in research, you end up having, there's a question at one time that needs to be answered, and you get into it. Mm-hmm. I was working on muscle disease before, and I wanted to look a bit more on neuro. When I started my postdocs after my thesis, the gene had just been identified, and I wanted to actually go to work on what's called post-gene, so trying to understand what genes do, can we make models, and can we go and find modifying therapeutic approaches. So that's why I started. Why am I still working on FA? Because we want to get this over with.
Sure.
And because it's family, so kind of, it's a large family now, so and I think there's still, you know, I think we can make a difference, and so that's why I'm still working on it. On your second question, in terms of, well, so we're both European.
International collaboration.
So international collaboration for me has always been the way to go. We were both at the first international meeting, which was not very, it was international, in 1999. It was a very few people.
Yeah.
I think we need to collaborate both with, I mean, this is a rare disease. If we each stay in our own little spot, in our own little town, I mean, we won't be able to get this done. So we need to have this international collaboration to share. And, you know, it's not because you're American or French or Italian or an Italian in Belgium that it's going to be different. It's still the same disease. And, you know, it's by bringing as scientists and clinicians, by bringing our brains together, that we can make a difference. And as patient and families of patients, it's by bringing what you know also about the disease together and working together that we can get this done.
Mm-hmm. Massimo, why FA?
Why FA? Well, my story is slightly different because I trained as a neurologist, and I trained as a neurologist in the early 80s. Okay, I got my medical degree in 1980. So at that time, most neurodegenerative diseases, all neurodegenerative diseases, I would say, were kind of black boxes. We have no idea why they come. I don't want an idea what to do with this. We just observe, make a very interesting diagnostic exercise. And of course, that's a state of the art that I didn't like, and nobody liked it. and there was a time when genetics was exploding, let's say, and the idea was that why don't we look at a genetic disease and try to figure out what is the problem, and then that may also be a model for, in general, for the study of neurodegeneration that is so obscure at that time. And why FA? For the simple reason that my mentor at that time was involved with FA, Stefano Di Donato. And the story of collaboration, and I just mentioned the fact that the discovery of the paper reporting the discovery of the gene, the corresponding authors from 2 different countries. I was in the US at that time, and had authors from 4 different countries. For your information, the United States, Italy, France, and Spain. So, to me, it has always been obvious that collaboration had to be the way to go with rare diseases.
Well, we're incredibly lucky to have you both in the field working every day on helping our friends and our families and our loved ones get to the end of this. So thank you again for tuning in. This has been a really interesting interview. They all have been fascinating. I hope you'll tune in again with our next series of interviews in the FARA interview series, Behind the Scenes at IARC 2019. Be sure to check us out on FARA's Facebook page, on FARA's YouTube, Twitter, and LinkedIn. Until next time, thank you very much.